How is that?
That's great.
Great.
Yeah, I think we're live.
Hey, hello everyone to the Early Career Immunology Seminar Series. My name is Dr. Anthony Cavruvius, and today I'm excited to host Dr. Jennifer Oyler-Yaniv from Harvard Medical School. Dr. Jennifer Oyler-Yaniv earned her bachelor's at St. Joseph's University in Philadelphia, where she also was a research assistant at UPenn studying the role host-pathogen interactions in pulmonary infection. Dr. Oyler-Yaniv went on to earn her PhD at Weill Cornell Medical College, working in the lab with Dr. Gregory Allen Bonnet. And the title of her thesis was How to Catch and Release Signaling Enables Long-Term Gene Expression Change in Response to Inflammatory Stimulus. Dr. Oyler-Yaniv then went on to UCLA, my home institute, where she did her postdoc in the lab with Dr. Roy Woolman, studying the effects of inflammation and how that determines the cell fate during viral infection. Since 2021, Dr. Oyler-Yaniv has been an assistant professor at Harvard Medical School. And today I'm really excited to hear her lab's current work titled, or her current work and talk titled Variability and Plasticity in Programmed Cell Death and what it means for immunotherapy and infection. So Dr. Oyler-Yaniv.
Yeah, thank you so much for having me. I think it's awesome what you guys are doing, trying to provide a forum for early career folks to share their work and to connect. It's just a great idea. So I'm really excited to be here virtually sharing some of the first work that's come from our lab. So this is also completely unpublished so far, almost all of it. I think there's only a little bit of it that's published from our postdoc. So excited to get your feedback and on what we've been working on. So what got me initially, first I should go with acknowledgement. So there's been one person in our lab in particular, the brave founder. We've grown since then, but Teddy was the first person to join our lab. He did almost all of the work that you're going to see today. And he's been just a great sort of initial person that we hired. So what got me into this problem initially was checkpoint blockade immunotherapies. So, and I think this is probably historical more than anything else. While I was doing my PhD I was working in a lab at Sloan Kettering, even though I was with Weill Cornell. And it was sort of the heyday of like the first clinical trial results coming out on the effects of CTLA-4 and or PD-1 blockade for patients with melanoma. And it was really exciting. A lot of the clinical trials were being conducted at Sloan. It felt like there was a tremendous amount of energy, and there was this real feeling that basic work on the immune system, on studying the immune system, was leading to tremendous benefits for people with cancer. And so it was a really energizing time, and I've always had an interest in checkpoint blockade immunotherapy since then. I don't really need to give this slide for this community, but the way that checkpoint blockade works, obviously, is by unleashing the patient's natural adaptive immunity against the tumor by antagonizing these inhibitory receptors, PD-1 or CTLA-4. And during this sort of early days of this sort of work, there were some interesting observations, and it's that it was the sort of core of that was that some cytotoxic treatments seem to be more effective in animals that had an immune system. As we all are familiar, a lot of immunocompromised mouse models are used for tumors, but it was found that in the context of an immunocompetent animal with a tumor, chemotherapeutic drugs seemed to work better. And the question was really, and the same later work showed that the same goes for radiation therapy. And the question was really why? And that there isn't an easy answer for that. There's a lot of answers as to why, but at least in part, it was discovered some cytotoxic treatments can induce inflammatory cell death. So let's sort of back up. Cells can die in multiple different ways. There are three kind of main types of programmed cell death: pyroptosis, necroptosis, and apoptosis. And these are controlled by distinct biochemical signaling cascades. I'm going to keep the same color scheme for each of these types of cell death throughout the talk. And you can see there are some gray arrows indicating crosstalk between the different pathways. So even though they have sort of distinct biochemical cascades, there's a lot of crosstalk between the pathways. And these different forms of programmed cell death culminate with differing morphologies, sort of characteristic morphologies. And what's important is that the mode of cell death causes different levels of inflammation. So both pyroptosis and necroptosis are thought of as inflammatory forms of cell death, whereas apoptosis is more anti-inflammatory and tolerogenic. And the reason for that is twofold. Pyroptosis and necroptosis culminate with lytic cell death, so the expulsion of the cell's innards into the microenvironment, and those innards contain DAMPs, damage-associated molecular patterns, as well as IL-1 family cytokines, which are very immunostimulatory. Apoptosis keeps the cellular innards encapsulated in these apoptotic bodies, thereby shielding them from the environment. And then phagocytes can come along and eat them. And the actual act of consuming apoptotic cells can enforce a more anti-inflammatory phenotype in macrophages. So getting back to this original question of why do some cytotoxic treatments work better in an animal with an immune system, the answer is that in some cases, those cytotoxic treatments cause inflammatory cell death. In this case, I'm showing pyroptosis and necroptosis, and they release DAMPs and cytokines, which can cause the maturation and polarization of antigen-presenting cells and T cells, and also generates antigens for T cells. And so there is clearly clinical value in pairing drugs that drive inflammatory cell death with checkpoint blockade-based immunotherapies. And as of 2020, there were at least 55 different clinical trials that paired drugs that induce inflammatory cell death with checkpoint blockade-based inhibitors. However, it's not always clear how a particular drug what type of cell death is driven by a particular drug. I want to just step back a second and show the evidence for this. A lot of this work has been teased out in animal models, but I think there's been one sort of really good human study that demonstrates this principle, and that's the tonic study. So in this trial, individuals were randomly assigned to different groups, and they were assigned doses of either doxorubicin, cisplatin, cyclophosphamide, or radiation, or nothing, and then given a checkpoint blockade therapy. And the key to this study is that they took biopsies of the tumor before, after the conclusion of the sort of immune priming drug, and then after, again, during the PD-1 therapy. And I'm just going to show one piece of data from that, which is that this is sort of transcriptomic data after the conclusion of each of the immune priming. And you can see that doxorubicin and cisplatin sort of preferentially induce this sort of inflammatory gene expression signature, as opposed to cyclophosphamide or radiation or nothing at all. And that correlated with a better response to the PD-1 therapy. So, as I mentioned, it's often unclear, however, which types of cell death are activated by a particular drug, and that's for a couple of reasons. There are some biological challenges, so there are differences across cell types. So a drug that induces inflammatory cell death in one cell type might not in another. There's also a lot of crosstalk between the different death pathways, as I mentioned. So if you're assaying for a particular effector molecule and assuming that it's sort of a bona fide it's a bona fide marker of how a cell is going to die, that might not actually be true. There's also a lot of heterogeneity in death pathway activation. So as I mentioned, there are differences across cell types, but even within a population of cells that's otherwise identical, some cells may die by one pathway and some cells may die by another. There's also sort of practical challenges. How cost-effective or scalable it is to determine how a particular cell is dying. And often the high-throughput approaches are done in bulk rather than single cells. So we have no idea about this sort of heterogeneity aspect. So just to demonstrate that principle a little bit more, we've shown that when you treat HT29 cells, that's a colon adenocarcinoma cell line in this case, with a particular drug, a common chemo drug, cisplatin, there are multiple different death pathways that are activated at the same time. So we can see evidence of cleaved caspase-3, which is canonically thought of as an apoptotic marker. And we also see evidence of phospho-RIP, which is canonically thought of as a necroptosis marker. And this is at the same time in response to the same dose of treatment. And the same goes when you look at all these different drugs as well, mitomycin C and paclitaxel. So different chemo drugs elicit sort of heterogeneous death pathway signaling. And when we looked at their morphology using sort of bright field microscopy, we also saw that different chemo drugs elicited a ton of morphological heterogeneity. So in some cases, you see formation of these little apoptotic bodies around a condensed nucleus. In some cases, you see this asymmetric nuclear condensation, cell swelling, vacuolization. And so we ultimately thought that perhaps, given the problems with assaying for sort of the molecular or biochemical evidence of programmed death pathway signaling, that maybe morphology will be a better way to assign how cells ultimately die. After all, it reflects sort of the convergence of a lot of different programmed death pathway and stress pathway signaling, as well as it sort of reflects what actually happens at the time the cell dies. Does it expel its contents to the microenvironment, or does it keep them encapsulated? And a good tool to do that is computer vision-based approaches. So let's use as an example these three little furry animals. So Google has mastered the fact that you can get onto your computer and you can Google a hamster and you get a hamster. You don't get dogs, even though these things are all sort of related. They're fuzzy, they have two eyes, they have a nose and two ears. They look similar, but it's easy to tell them apart. And computer vision is shining already in search engines and things, and really all over the place, to perform this task of classifying different things that are different entities. And so given that apoptosis, necroptosis, and pyroptosis look very different, we thought that we could apply these computer vision-based tools to distinguish how cells are dying using just morphology alone. So in this image, what you're looking at this is a different cell type, not our colon adenocarcinoma cells, but these are just plain 3T3 fibroblasts that are treated with ligands that preferentially induce one particular type of cell death. And you can see that they really look different. And so that encouraged us that we might be able to use this technique to classify how cells die. So to do that, we trained a neural net to distinguish cell death modes based on morphology alone. And the way it works is like this. So we take a population of cells. In this case, again, we're using our colon adenocarcinoma cells. We treat them with compounds that uniformly induce one particular type of cell death. To induce apoptosis, we treat them with Tnf alpha, cyclohexamide, and necrostatin to block RIP signaling. To induce necroptosis, we treat them with TNF, cyclohexamide, and ZVAD, which blocks caspases. And to induce pyroptosis, we use lipopolysaccharide and nigericin. We then image the cells over time, and when they're treated with these particular death-inducing compounds, take pictures of individual cells, and then we train our convolutional neural net using these images, single images of individual cells just prior to the loss of nuclear membrane integrity measured by, as we measure by dye incorporation, like a standard cell death dye. And then the idea is once we have a trained classifier, we can then use that classifier in situations where we don't know ahead of time how the cells are going to die. We can say this morphology is indicative of the cell dying by apoptosis, this morphology is indicative of cell dying by necroptosis, and so on and so forth. So how does it work? I'm first just going to show you some examples of how our cells look when we treat them with our sort of prototype apoptosis, necroptosis, or pyroptosis inducing drugs. You see that in the context of apoptosis, you see nuclear condensation. There's a lot of cells dying in these images, but I'm highlighting a few just for sort of simplicity. See that we get nuclear condensation, cytoplasmic shrinkage, plasma membrane blebbing, and then this ultimately ends up with plasma membrane rupture after a few hours. That's secondary necrosis, which happens more often in vitro as opposed to in vivo, where a macrophage would come along and gobble these cells up before they would lose membrane integrity completely. Necroptosis, by contrast, looks very different. The cells round, they lose cell-cell contacts, they swell, and then the plasma membrane abruptly ruptures in this very spherically symmetric way. Pyroptosis is, again, still different. You often get sort of asymmetric nuclear, the nucleus moves to one side of the cell, you get vacuolization, the nucleus doesn't expand quite the same as necroptotic cells, and then you often get cell swelling and plasma membrane rupture that is asymmetric as opposed to necroptosis, where it's often spherically symmetric. So how does our, I should also say we stained for the signaling pathways that are sort of canonical for each prototype form of death. So I stained for cleaved caspase 3. In the case of apoptosis, we see the apoptosis promoting conditions, we see evidence of cleaved caspase 3, specifically surrounding these cells that have condensed or fragmented nuclei, which is sort of indicative of apoptosis. When we induce necroptosis, we stained for phospho-RIP, and we see widespread phosphorylation of RIP without this sort of same degree of nuclear condensation and fragmentation that we see in the apoptotic condition. And for pyroptosis, we first stained for this molecule AIM2, which makes the cells sort of competent to undergo pyroptosis, and we see that they're expressing high levels of it. And then when we stained for the presence of NLRP3 sort of speckles after treating with our pyroptosis promoting conditions, we saw that we get this formation of these little speckles after we've treated with the ligand, right? And I'm not going to show you for the sake of time all the other data, but we know that when we treat with these prototype sort of drug cocktails, we don't see evidence of the other forms of cell death at the level of signaling. So we also see that when we treat with these prototype cocktails, we get the elicited sort of pattern of DAMP release. So ATP release, extracellular ATP is indicative of lytic cell death. And we see that at about 6 hours post-treatment, necroptosis and pyroptosis promoting conditions, we see much more accumulation of extracellular ATP as opposed to apoptosis, which is consistent with what we would think when those membranes are still intact in our apoptosis conditions. So how does the classifier perform? For those of you who are interested, so it has sort of high level of precision and recall. So just to be clear about how we did this, we trained our classifier and then we kept some of the data back during the training process. And then we tested the performance of our classifier using that data that we'd held back. So this classifier had never seen this data before. And we see that it has an overall accuracy of about 94%. We can then look at a confusion matrix and see when it messes up, where does it mess up? So this is the predicted label, what the classifier, how it thinks the cell died. And this is the true label. So how it died as opposed to based on what drugs we gave it, what prototype drugs we gave it. So the top number represents the number of cells in this particular experiment that it classified, and what's the proportion of those cells that it called correctly. So we can see that for the most part, it's performing fairly well for all three classes, and there's not really a systematic or bias in the sense that we don't often see one type of cell death getting incorrectly called as another type. It seems sort of distributed. So now we can take our trained classifier and put it in and see how it does when it's confronted with data from images where we don't know ahead of time how the cells are going to die. So to do that, we took our HT29 cells and we treated them with varying cytotoxic compounds, and we see sort of modest cell death across these different treatment conditions. And one of the things we found was that when we treated the cells with these different types of conditions and then measured ATP release, which remember is indicative of lytic or inflammatory cell death, we found that this sort of amount of ATP that's released doesn't correlate almost at all, or it's poorly correlated with the amount of total cell death. And that indicates to us that the amount of ATP is poorly predicted by the amount of total cell death, and is instead probably dominated by some hidden variation in the way that the cells are dying in response to these different treatments. And when we applied our classifier to these different compounds, we saw that there's a lot of variability in how the cells die. So in response to TNF alpha, most of the cells are dying by apoptosis, whereas when we treat the cells with paclitaxel or EGFR inhibition, we see that most of the cells are dying by necroptosis. Now, because pyroptosis is the least common form of cell death across all of our conditions, which isn't surprising, it's canonically thought to be downstream of microbial signals, we wanted to confirm that our classifier was accurately calling this type of cell death. So we included a different inducer of pyroptosis. In this case, we used LPS and monosodium urate crystals. We saw that in this case, most of the cells that die are assigned to pyroptosis. So we're confident that our classifier is performing reasonably well for that class of cell death as well. This time, when we correlate the amount of ATP that's released with the amount of lytic cell death, necroptosis and pyroptosis, we see that the correlation improves dramatically. You can see that EGFR inhibition which was dominated primarily by necroptosis and pyroptosis, releases much more ATP than, say, TNF alpha. So let's look a little more carefully at these two types of cell death, EGFR and TNF. They're really at the far ends of the spectrum in terms of ATP release and necroptosis versus apoptosis. So when we looked at these, we looked for other markers that are indicative of inflammatory cell death. So in the case of TNF alpha, we see that it only sort of modestly induced LC3, which is a marker for autophagy. It increases its expression modestly over the control, no treatment condition. You see some of the sort of prototypical granules that are associated with sort of LC3 condensation. Whereas when we've treated with EGFR inhibition, we see massive upregulation of LC3 expression and formation of these sort of LC3 clusters. This is consistent with evidence that from papers that are shown down here in the left-hand corner, that autophagic signaling is required for ATP release in cancer cells. We also saw that EGFR inhibition, as opposed to TNF alpha treatment, is associated with the loss of the nuclear damp HMGB1. So HMGB1 is normally present in the nucleus, which is where you see it primarily for this sort of controlled treatment, and TNF alpha treatment, where it's sort of tightly present only in the nucleus. Whereas when we treat with EGFR, we see that there are these regions where there's no green signal at all, just blue nuclei. And when we zoom in on those a little bit more, you can see that the nuclear signal for HMGB1 is lost, indicating that this damp is getting released by the cell. So we next decided that what would be great to use our classifier for is to screen for potentially immunogenic drugs. And with the idea being that it's a pretty cost-effective, efficient, and easy, honestly, way to identify drugs that may induce preferentially immunogenic or inflammatory forms of cell death as opposed to apoptosis. And the principle is just that we would conduct an imaging-based screen of a modest number of compounds, and then we'd apply our computer vision-based classifier and ID for potential hits, with the idea being that if we find, in this case, this cartoon, drug B, almost all of the dead cells are undergoing apoptosis, so that would be a potentially tolerogenic drug to be avoided with checkpoint blockade therapies, whereas drug C, you're seeing a predominance of necroptosis, might be a better drug to pair with immunotherapies. So we screened a library of about 160 different cytotoxic compounds targeting a range of different sort of molecular targets. And we saw that there was variable overall cell death. So some of these compounds in sort of scorched earth killed almost every cell in the dish, and some of them only induced about, 10 to 20% of the cells to die. But when we applied our classifier, we also saw a lot of variability in how the cells were dying. So each of these points represents a single drug. And you'll see that some of the compounds preferentially or almost solely induced apoptotic cell death, whereas some of the drugs induced necroptotic cell death. And we're only showing this on two axes because if you'll recall, pyroptosis was very uncommonly assigned. And that's reflected also here, where most of the drugs lie on this diagonal and only a few out here were being assigned as pyroptotic in addition. So right now, what I can tell you is that drugs up in this corner seem to preferentially be HDAC inhibitors and mTOR inhibitors, whereas drugs down in this corner that are inducing necroptosis seem to be PI3 kinase inhibitors. I want to, be clear that I think that this might be the real strength of our screen is that we don't think every cell line is going to die in the same way in response to the same drug. So maybe colon cancer cell lines die and have these responses to these different types of drugs, but maybe if we tested another cell line, it would be different. And we think that's where our technology sort of shines, is that it's flexible and it's easy to apply and fast for a lot of different cell lines. And so this gave us an idea, and we started wondering if maybe the best chemo drug for combination with checkpoint blockade might be a mild killer. A lot of these, or even a killer that's more specifically targeted to tumor cells as opposed to a more general chemotherapy. These are often associated with really bad side effects for the patient. Maybe we could find a drug that its only purpose is to really promote a microenvironment in the tumor that is conducive to the adaptive immune system functioning. And then we could spare the patient some of the really nasty side effects of chemo, or preferentially target only the tumor, or even use just a drug that doesn't even kill that well, might be a good drug to pair with chemo. So let's take as an example and compare Tnf alpha, which obviously isn't chemo, but as a proof of principle sort of to this idea, let's compare Tnf alpha and EGFR inhibition, which again, in this little panel of drugs they're at the sort of far ends of the spectrum, right? So TNF is killing about more than 2 times the rate of EGFR inhibition. But if we return back to this panel, we see that they're also at the other ends of the spectrum in terms of how they're killing the cells. So EGFR inhibition promotes necroptosis as opposed to apoptosis. And when we look at how much ATP is released, again, EGFR is far more than TNF alpha. So it might be promoting a much better environment for, say, checkpoint blockade-based therapies. And again, TNF is not a chemotherapy. Nobody's using that in patient, but it's just sort of to prove this principle. So I'm going to just summarize this part of the talk. We found that chemotherapy drugs kill cancer cells in variable ways. We learned this by looking at death pathway signaling, using immunostaining and assaying for the presence of DAMPs released by dying cells. We think that morphology might be a really great way to assign cell death and get around some of the problems, both biological and practical, that come with trying to determine how cells die. And this work is inspiring us to try and think of different ways that we could identify good cancer drugs to pair with checkpoint blockade-based immunotherapies. So that part of the talk was obviously all about immunotherapies and cancer, but I want to switch gears now and change to sort of our host pathogen wing of the lab. And one of the things that we were thinking about was the simple question of why do cells have all these different ways to die? When you think about proliferation, there's only one cell cycle. There's a lot of stuff that feeds into it, but there's only one way to divide, but there's a lot of different ways to die. And one of the reasons for that, or at least the sort of hypothesized reasons for that, is to protect against viruses. And the way that this principle works is simple. Imagine you have a healthy or susceptible cell and it gets infected with a virus. if that cell dies before the virus can complete its life cycle, then it can't spread to neighboring cells, right? And so the hypothesis, which I think is there's a lot of evidence for it at this point, is that having multiple death pathways provides redundancy and robustness to viral interference with cell death pathways. And I think that the fact that these types of cell death are associated with inflammation and the release of IL-1 family cytokines and DAMPs that stimulate the adaptive immune system is further evidence for this, the likelihood that this hypothesis is true. In addition, viruses have evolved tons, not just viruses, bacteria too, have evolved tons of different mechanisms that antagonize the components of programmed death pathway, which indicates that it's important for them to block the cell from dying. So all of this argues that the death pathways evolved in part specifically as an antiviral mechanism. So I'm going to return now to some data that's already published. We found that treating cells with the cytokine Tnf alpha increases the death rate of infected cells, which then restricts viral spread. So what you're looking at is the rate of death for cells that are treated with different doses of TNF, and these are cells that are infected with the virus HSV1. And you can see that it just increases how fast the cells are dying. And this corresponds with a reduction in viral spread. So you're looking at the same cells, the percentage of cells that are infected with the virus at 48 hours post-infection in response to these different doses of TNF. And we know that TNF alpha's protective effect is mediated by cell death. And we know that because when we add drugs, I've mentioned these before, Zvad or necrostatin, which block caspases or RIP1 kinase, we can completely abrogate the protective effect of TNF alpha. So the way that TNF confers its antiviral effect is by increasing this arrow. It increases the probability and the rate at which infected cells die, which thereby reduces the likelihood the virus can complete its life cycle and spread to neighboring cells. So one of the things you'll notice is that it took 2 drugs to abrogate TNF's protective effect. But what happens when we add just the drugs by themselves? So you're looking right here at images of the cells infected with HSV1. HSV1 in this case is tagged with M cherry, so we can visualize it pretty easily. And at 48 hours post-infection, you'll appreciate that only some of the cells in the population are infected with HSV, whereas when we've treated with no TNF, the whole plate is infected. Every cell that's there is infected with HSV1. When we add just Zvad to block caspases, we see that TNF can still prevent the virus from spreading. Death is still sufficient to prevent viral spread in culture. The same goes if we add just necrostatin 1. Again, TNF can still protect against viral spread. But when we add both drugs together, then death is no longer an effective mechanism to prevent the virus from spreading throughout the population. So what this all means is that flexibility in the death decision confers robustness to perturbations. In this case, the perturbations are these drugs that we've applied. We have to really block everything in order for the virus to be able to spread in culture. So what this indicates is that cells in a dish, and we've switched at this point, I should have said this earlier, these are no longer HT29 cells, we've switched to 3T3 fibroblasts. What this means is that this particular cell type is competent to undergo different types of cell death in response to this virus. And this is confirmed when we look at death pathway activation during infection at the molecular level. So what you're looking at in these images are HSV1 infected cells. In this case, we've stained for HSV1 ICP4. And then in each image, we've stained for a different death pathway effector, cleaved caspase 3, phospho-MLKL, phospho-RIP, pyrin, or we've applied an isotype only. And you can see that in these infected cells, preferentially, not the uninfected cells, we see evidence of activation of each of these 3 major death pathways. which raised the question of, are these single cells at the single cell level, does a single cell activate only one death pathway at a time, or does it activate multiple death pathways simultaneously? So we did some additional immunofluorescence where we stained for multiple death effector proteins simultaneously and saw that at the single cell level, for single infected cells, demonstrate staining for each of the three major death pathways: cleaved caspase 3 apoptosis, phospho-MLKL necroptosis, and ASK in this case as evidence of pyroptosis. So I think this shows sort of further evidence for this idea that flexibility in the death decision confers robustness to the cells against potential perturbations by the virus in blocking components of death pathway signaling. So we decided then, in addition to sort of, this is cool, it also makes it really difficult or impossible for us to use staining to indicate how a cell ultimately dies, right? Because the virus-infected cells activate all the different death pathways. So we don't know just by looking at, say, phospho-RIP staining that the cells are going to die by necroptosis. So we decided to use our cell death classifier to gain insight into how these virus-infected cells ultimately die. When we look at bright field images of the cells that are infected with virus, you see that there's a lot of morphological heterogeneity. We see vacuolization here. There's more vacuolization. We see apoptotic bodies here. We see a lot of cells that just round up and then swell and explode. These are just going to show you a few examples of these sort of morphological heterogeneity present in this dish. And so we retrained our neural net to distinguish cell death modes for 3T3 cells as opposed to our HT29 cells, which we used earlier, which I was sharing with you earlier in the talk. The principle is the same. We take our 3T3 cells and we treat with compounds that uniformly induce one type of death. We acquire our images and then train our CNN using the images of the individual cells. And then we can go back to our plates where our cells are infected with viruses and are dying in a lot of different ways, and we can classify how the cells are dying based on their morphology alone. This is just to indicate to you that our classifier performs well when retrained on dying 3T3 cells. So we have an overall accuracy of about 90% and reasonably high precision and recall values. And in addition, when you look at the confusion matrix, in this case, it was sort of smaller experiment, so there are fewer cells that were tested. And we see there's also not really a systematic error, maybe between apoptosis and necroptosis, but it's not, doesn't seem too bad. So when we then look at the Logit scores, which is how the cells are ultimately, how the classifier assigns cell death, we see that we get clustering of the sort of necroptotic, pyroptotic, and apoptotic cells in these three different wings. And we see that there's just this, the HSV infected cells sort of occupy this whole space. So we see some cells dying by apoptosis, some by necroptosis, some by pyroptosis. And this is reflected in this pie chart where you're seeing that about 35% of the cells in this case were assigned apoptotic, 43% pyroptotic, and 23% as necroptotic. And I think we're still trying to figure out how to sort of how to make sense of this data, because it seems like there's also just some, if for lack of a better word, confused forms of cell death. So I don't think that cells need necessarily adopt one type of cell death, but they might exhibit evidence of multiple types of cell death acting simultaneously, or sort of intermediate forms of cell death. Again, we're still trying to figure out how we could show that using sort of morphological analyses. It's not easy using the data that comes from the classifier alone to do that. So if you're looking at this and you're like, well, that's sketchy. She's getting like relatively equal proportions of cells undergoing apoptosis, necroptosis, and pyroptosis. So how do you know that your classifier isn't just sort of randomly assigning cells into each category? What we did in this case was we took advantage of a particular useful feature of the 3T3 cells, which is that when we know from a lot of other experiments we've done with them that when we treat them with mitomycin C, they very exclusively activate apoptotic signaling as opposed to anything else. So mitomycin in 3T3 cells, at least, strongly induces apoptosis. We can see that they show evidence of cleaved caspase 3 staining, cleaved PARP, I'm not showing it, but cleaved caspase 8, and no evidence of phospho-RIP, phospho-MLKL, or ASK. So we know that they don't activate any of the other programmed death signaling pathways when we treat with mitomycin C. So then we can go and image the cells that are treated with mitomycin C and see how our classifier performs on them from morphological analysis. So when you look at how these 3T3 cells are dying in response to mitomycin C, you just see tons of nuclear condensation. You see all these little, the dish is just littered with apoptotic bodies, which is consistent with what we know about the sort of signaling that is happening in these cells at the same time, that they're only activating apoptotic signaling. We see that they're also by eye activating pretty exclusively apoptotic cell death. And so when we use our classifier, we can look, this case we're clustering again, our prototype drugs or prototype forms of death. So pyroptosis, apoptosis, and necroptosis, these are cells that are treated specifically with the drugs that we know induce only one type of cell death. And then when we overlay how the cells are dying when we've treated with mitomycin C, we see that they cluster preferentially on top of the apoptosis conditions, and upwards of 80% of these cells would be assigned as apoptotic. So that gives us confidence that our classifier is accurately discerning cell death when we've infected them with the virus. So to sort of summarize that part, we found that single virus-infected cells promiscuously activate death pathways and execute diverse types of death. Single cells show evidence of activation of sort of the molecular effector proteins associated with each type of these major forms of cell death, apoptosis, necroptosis, and pyroptosis, and seem competent to ultimately execute each of those types of death. This means that in some contexts, cells are flexible in which death fate to adopt. We think that this is a mechanism that could confer robustness to interference with host cell death. So viruses that try to block one component of cell death signaling, the cells have already activated all these other forms of cell death, and they have many avenues to still die in the face of these sort of viral perturbations. And we're starting to think about if there are ways that we could preserve the antiviral qualities of cell death while reducing the associated inflammation. So are there ways that we could treat virus-infected cells with drugs and shunt the proportions of cells that are dying by inflammatory versus anti-inflammatory death pathways? And in contrast to the first part of the talk, where we maybe want to promote inflammatory cell death, in the context of viral infection, rampant inflammatory cell death might actually be detrimental to the host and spur sort of hyperactive immune response. And so maybe we could shunt cells towards apoptosis rather than necroptosis or pyroptosis. Those are things we're still trying to explore. And with that, I will acknowledge everybody who's involved in the work. My partner, Alon, and I run the lab together, so this is really a joint effort. Teddy is the one who did almost all of the work that you saw today. We would be nowhere without generous sharing of reagents from David Knight's lab at HMS and Prashant Desai at Johns Hopkins. They've shared essentially all of their viruses and know-how with us. And all of this work was made possible by the Nikon Imaging Core Facility at HMS. And so with that, I will take any questions that anyone might have, and I'll also stick around afterwards to chat with whoever joins the Zoom there.
Yeah, really awesome talk. I really enjoyed it.
Thank you.
Totally, Yaniv. Yeah, I had a question about, I thought it was really remarkable, and your data is really cool, showing the different fates that cells have, even in regards to the same stimuli. But one question I had is, it looks like if you perturb the same pathway, you get different results. And what I mean is, like, you showed very nicely that the PI3 kinase AKT mTOR pathway, we kind of think this is as kind of a linear pathway, but depending on what component you target, you get a completely different result from apoptosis to necroptosis. So I thought that was really fascinating. I always thought if you block the same pathway, you would generally get the same result, but that's not the case.
Same. So that I actually think away from the practical sides of things, it might be also a very useful way for us to learn about the mechanisms that control each, like to learn about the basic biology of what's controlling the cell fate decisions. The mTOR pathway was one that was in particular very hard to get our head around completely, because in some cases you could treat with a drug that's like it's like let's say temsirolimus versus everolimus. They're like only derivatives of each other, but in those two drugs we see like very different. So while let's say mTOR inhibitors preferentially induce apoptosis for the most part, there are some of the drugs that induce necroptosis preferentially, and there's not always a clear sort of structural predictor in the drug that we know like that will induce it this or versus another. The cisplatin or the platinum compounds are similar. Like some of them induce inflammatory death and others don't, even though you'd think that they'd all be the same. They're not. And so I think that it might be an interesting way that we could learn about really the basic biology of what controls death fate commitment in a particular in cells, right? Just by looking at the effect of our drug screen.
Yeah, that's really cool. So it suggests there's even like a rheostat depending on how potently you inhibit the same target. Are you guys also controlling to make sure there's no off-target effects of some of these like rapalogs?
No, I don't know how to do that. It's possible there are it's not possible. It's likely that there are off-target effects that are happening. One of the things that I think would probably be important to do at some point is to go back and test some of the drugs again with different doses and see that maybe it's possible that once you reduce the dose, that some of the off-target effects would stop happening and you'd see different forms of cell death predominate when you take down the dose. Yes, but the answer to your question is no, we're not really controlling for that yet. And there are probably a lot of off-target effects that we have to deal with.
Okay. Yeah, that's fascinating in regards to the dose, because that may be relevant for the clinic, right? Depending on how much dose a patient receives may tip that balance from apoptosis to necroptosis or pyroptosis or vice versa.
Yeah, so. Absolutely. And just the pharmacodynamics of how much get into the actual tumor, which is always like way lower, right, than what the patient gets usually infused or something.
Yeah, correct.
Yeah.
Okay, I'm going to go to the YouTube chat section and just take some questions. So Shu asks, in the apoptosis versus necroptosis plots, are several drugs are off the straight line? Do these drugs trigger more pyroptosis, or do cells begin to have resistance to them?
Yeah, the ones that are off the straight line have been assigned. There are also a pyroptosis assigned to them. We're just not showing it in that particular plot because most of the cells don't undergo pyroptosis in response to any of the drugs. There's probably also a couple of them that are just they don't fall into any of the classification of the 3 classification schemes.
Okay, Adiba Azad asks, some subsets of T cells portend a more poor prognosis if present in tumors. Is there evidence of priming of certain T cell subsets to specific forms of cell death?
Well, I guess I can't ask you if they mean priming the T cell to die or priming or the T cell's response to the dying cells. I suppose I will answer both of them depending, like, just in case it was one or the other. So I don't, in terms of priming the T cells themselves to die in different ways, I have no idea. We haven't done anything like in primary cells or in T cells yet. But in terms of the question of like, do cells that die in different ways preferentially prime different subsets of T cells? I think it would be an interesting thing to figure out. A lot of the work, the way that we're following up on this is trying to look downstream and asking, what is the effect of these dying cells on immune cells? We are trying to look at their effect on both sort of macrophage populations, seeing how they change, their phenotype changes in response to the stuff that gets released by dying cells, but also co-culturing with T cells to see how it affects sort of T cell function. T cell subsets is a harder question because you often need to move in vivo to do that. And we're still pretty far away from doing that. But I think it's a really good point and a good question because that's obviously very important for the outcome of the tumor too.
Yeah, I think that's really important. And I think you mentioned that or alluded to earlier in one of your figures where you mentioned like certain forms of cell death may promote a more robust inflammatory response or even more tolerance to the tumor. So yeah, really cool. I look forward to future work in that area.
Thanks.
Okay. George Sue said, great work. Have you checked caspase-1, GSDMD, and IL-1 beta cleavage by Western blot and HT29 cells undergoing apoptosis or pyroptosis?
Yes. We have not looked at cleaved caspase-1, but we have looked at gasdermins. And do see activation of the gasdermins. What was the other one he asked?
IL-1 beta and caspase-1, so essentially the inflammasome components.
Yes, IL-1 beta. We've measured IL-1 beta by ELISA, not by Western blot. I have not done any Western blot experiments, or Teddy has either with the HT29s, but we have with the 3D3s and confirmed sort of more widely the activation of the different death pathways, which, as everyone who works with antibodies knows, there are many more available to you when working by Western blot as opposed to IF. Seeing something in the chat.
Okay. Yeah. Shu asks, do you think the classifier of cell death needs to be trained for each cell type? Possibility of recognizing types of cell death in tissue sections.
Yeah. It's a great question, and everybody asks me this. So right now, it always needs to be trained on every new cell type. I think it's possible that as we increase the sort of space of different cell morphologies that we see in our classifier, that it might be able to perform well, despite differences in the underlying sort of cell type or cell morphology. But we're far away from that being possible yet. Right now, it really needs to be done in a dish. And so many people have asked me about whether it could work in a tissue section that I feel like we have to find a way to do it. I think there are in order to do that, so we need to generate a training set where we know how the cells are dying ahead of time, which we could do, but I don't know a good way to do it aside from just human annotation based on sort of signatures that we know are associated with each type of cell death. And that's not super scalable or fast. It's like clicking and being like condensed nuclei, cleaved caspase staining. Let's assign that to apoptosis and then generating the training set that way. That takes a long time to generate a big data set that will robustly train the classifier. But I think it could be something that's very useful for a lot of people. So maybe we'll have to find some clever way to do it. The advantage of our training scheme is that we don't have to use a human. We know that we can force cells to die in given ways, and then we have a big training data set. So that's sort of the advantage there.
Okay, sorry, 1 second. Yes, I think that's it as far as the YouTube questions. I have one last question before we go. So, you mentioned the 3 paths that these like, for example, infected cells have in regards to cell death. Is there a 4th option? I've studied cellular senescence, and there's evidence that viral infection may cause senescence. I'm not sure if that's something you're able to see in your own experiments, and whether you would be able to identify, use your algorithm to even pick up senescence in addition to cell death.
Yeah. So the short answer is yes. Senescence is always associated with this like really dramatic morphological change with the cell. It gets really big and flat and like spread out, like it enlarges, right? That's what I've always seen. So anything that's really visible by eye that you're like, can call a cell state or fate change by eye, definitely you could train a classifier to pick that out. And I think there actually have already been groups that have trained neural nets to distinguish senescent cells. So we wouldn't even be the first to do that. But I think it'd be really useful, right? Because then you don't have to do those annoying beta gal stuff that most people usually use, and I've heard are kind of annoying. But yeah, I think the advantage of this is that it's need not be, it could be expanded to include a lot more of cell state or fate, aside from just these 3 types of programmed death that we've started with.
Yeah, that's really awesome. Well, I think that concludes your the portion here in regards to questions into your talk. Yeah, really fascinating work. I think your lab's off to a really awesome start, and I look forward to seeing all the different veins you follow in your publications.
Thank you so much.
Yes, I think with that, we'll end the YouTube portion of the talk, and then we could go offline.
Okay. Okay. Now the only people, YouTube is off, right?